Good morning. So before I get into the talk, I wanted to go ahead and lay a little preamble here. So we're talking about evolution of protein interaction networks. The phenomenon I'm concerned with here are the fates that happen to gene duplicates. And there are three possible fates. One is non-functionalization, where given those two genes in the genome, one is going to be mutated and silenced, and that's the last I'm going to say of that. What I'm really concerned with are those gene duplicates that survive in evolution. And so survivors, there are a couple possibilities that have been posited. One is neofunctionalization, and the idea there is that these initially identical gene duplicates are going to be functionally redundant, and therefore one of them will be relieved from selective pressure, and the mutations that it acquires gives it some new, heretofore unseen beneficial function. So the neo and new function. Subfunctionalization came out of population genetics. The idea there is that these two duplicates are each going to assume some partitioning of the ancestral functions. So a portion of them will go to one gene and a portion to the other. Now, as we shift into actual protein interaction networks, this slide is the remedial I would really want to draw your attention to is on the right there, when you have two or more proteins that are interacting with each other, either homodimers, trimers. How that's going to be represented in the graph is going to be that node representing interaction coming back in on itself. Also representing gene duplication in protein interaction networks is fairly straightforward as well. The way you would do that is just there, you would make a copy of that protein that's expressed from the gene duplicate, and you'd copy its interactions as well. Now, neofunctionalization and subfunctionalization each have really nice analogs in protein interaction network evolution. And the idea here is that gene function is synonymous with protein interaction. So proteins with identical interacting partners putatively have identical functions, at least in the simplification of these evolving models. And as you can see here, after duplication, the two fates, neofunctionalization, the acquisition of a new function is followed with a new interaction between two proteins occurring that hadn't occurred before, a de novo interaction. And its complement down on the bottom, subfunctionalization, is that those interactions are then partitioned between the two duplicates. And so what I'm concerned with in this talk is neofunctionalization. There are a number of lines of reasoning that have come to the conclusion that yes, indeed, this happens quite a bit in evolution, and that's precisely what I want to take on in this talk. So there are three lines of reasoning that I'm going to take a look at. The first is the entire network is evolving. These are a little cryptic, but they become quite straightforward once we get to the end of the portion of the talk. So he and Zhang made an observation about subfunctionalization, and that is that if subfunctionalization is the only thing that's occurring in the network, what you're going to expect to see is that as those interactions are partitioning between expressed genes, the duplicates, that the number of interacting partners between those duplicates is not going to change. And what you would then expect to see is that if you group all the duplicates in your interaction network roughly by age, you should see no correlation between the age of those duplicates and the number of neighbors that they have. But in fact, they found that that wasn't the case at all. They found that indeed, the older the duplication, the greater the number of interacting partners that those duplicates had. And so, you would, as shown here, you would expect the number of neighbors increasing over time. And the explanation that they provide for that is that after subfunctionalization occurs rather quickly after the duplication event, that over time what's happening is you're getting neofunctionalization, these de novo addition of interactions that didn't exist before. And it's here's another look at it. So if this is your extant biological network, or a little subnetwork, and if you wanted to posit how it came to that form, you might have had this particular gene duplication occur, and then new interactions being added over time. However, getting back to that initial bullet point, the entire network is evolving. Okay? Those duplicates that you're looking at aren't the only thing that's evolving in your network. Let's look at this scenario. After the initial duplication, one of the interacting neighbors of that duplicate pair also has a gene duplication event, and you lose some interactions due to subfunctionalization. And then another neighbor duplicates later on in evolution, and you also lose some interactions due to duplication. Whoops. Yeah. And so what you end up with are identical outcomes for how that extant network came into being. And in fact, there are other lines that we looked at to validate this. So for instance, you'll find that that paralogs are indeed enriched for other paralogs as far as what they're interacting with. I would say that of everything, all these lines of reasoning that we looked at, this is probably what I think is the most significant, simply because it has possible ramifications in other areas of evolutionary analysis. Whenever you're pairwise studying something and you're making some assumptions or conclusions on how those two things that you're looking at, without attaching any meaning, whether they're sequences or something else, are you considering that everything else in the organism is evolving at the same time and may be affecting what you're seeing? So I have a lesson to be learned from this. So moving on to the second point, assays, missing one and one missing. Wagner came out with a couple papers also looking at neofunctionalization and studying its presence or absence in the evolution of protein interaction networks. And he made the observation that if you have a paralogous pair of proteins and they're interacting, there are only two ways that that interaction could have formed. The first way is that the ancestral protein could have been self-interacting, and then upon duplication, those initially identical duplicates would be interacting with each other, and then that interaction would have been preserved in evolution. The second possibility is that that ancestral protein was not self-interacting and duplicated, and there was initially no interaction between them, and then an interaction formed de novo. Well, looking at the data, he did a count and found that there were 34 pairs of proteins, paralogous pairs, I should say, that were interacting with each other, but were not themselves self-interacting. And so one of two possibilities had to explain this interaction. Either 34 interactions formed de novo in the network sometime over evolution, or there were these duplications and 68 self-interactions were lost. And obviously the more parsimonious explanation is the 34 de novo. And so then this number was extrapolated, and it was found that actually neofunctionalization is quite ubiquitous in the evolution of these networks. Well, in order to take a closer look at this, what we need to look at is the actual data. And where is all this majority of data that we're using in constructing these networks comes from large-scale assays, and there are two of them. The first is the yeast 2 hybrid assay, and this is a nice kind of assays 101 textbook picture of the yeast 2 hybrid assay. You have the Gal4 binding domain attached to a bait protein. It's bound to the DNA, and then a prey protein comes along with the activation domain attached to it. And if those two proteins interact, you get expression of a reporter. Well, what you don't see in these cartoons is that, in fact, that Gal4 binding domain binds the DNA as a dimer. So if we go back and look at our initial cartoon, actually is a more accurate representation of this cartoon is to show that binding domain as a dimer. Now, let's look at a scenario. What if that bait protein that you're trying to find its interactions, what if that bait protein is self-interacting? There's a propensity for it to interact with itself right there with the dimerized binding domains, and then there's no reporter to bind to get expressed. Let's look at the other large-scale assay, and that would be affinity purification mass spec. So a protein complex is isolated. It's exposed to a digest. You take all the peptides, run it through the black box. You end up with some mass charge spectra, and then you go ahead and you submit that to a database, and it comes back with the proteins that are the components of this particular complex. The problem here is it tells you what proteins are in the complex, but it doesn't actually tell you if any of them are binding as dimers, trimers, tetramers, and so forth. So in fact, if you look at the mass spec studies, you'll find they report no homomeric interactions. There is a variant on this where a flag epitope tag is used, and in those cases, you can actually differentiate, but that epitope tag is so small that in the mass charge spectra, it's very hard to detect. And in that particular study, in over 1,000 interactions reported, exactly one of them was reported as being self-interacting. So what we're finding with these large-scale assays is there's a real dearth of self-interactions being reported. So if we return back in the study and ask the question again, what's more likely is that there are actually quite a few self-interactions that we just simply aren't seeing from these large-scale assays, and the homomeric duplication is a much more parsimonious explanation for the formation of these interactions than neofunctionalization. I should also say that this idea is well supported in other types of studies, such as structural studies and so forth, and enzyme databases, which actually show an extremely large number of paralogous interactions and homomeric interactions in them. So that takes us to the final one, randomly added interactions. What do you expect? What this is getting at are the theoretical models that are used to promote ubiquitous neofunctionalization. And let me take a moment to explain these two plots. What's being looked at here are models of protein interaction network evolution. Both these models feature gene duplication. They both feature subfunctionalization. If you look at the x-axis at the bottom there, this is the probability of any redundant pair of proteins actually, or not proteins, any redundant pair of interactions actually being lost. And the way to group these lines is to group them by color. So each color pair represents a different parameterization of the model, and the solid line represents a run of the model itself, and the dashed line represents the equivalent network where you, in essence, preserve the degree distribution and randomize the edges on it. And what we're interested in looking at here is how are these models dealing with the clustering coefficient. And the reason the clustering coefficient is interesting to us is that it's generally accepted that the clustering coefficient is going to be much higher in a biological network than it is in a randomly generated network. So what you see is a model that's going to be able to show a nice differentiation between the model representing protein interaction network evolution and a random equivalent. Now, let's focus on the left plot first. The left plot is the additional mechanic being added here is neofunctionalization. So after you deal with the gene duplication, the subfunctionalization, now you're going to have some random edges. And that beta parameter there is representing on average the number of random edges being added with each duplication. Black line is 0, so this is the default model, and it's identical in both plots. But you can see at very low levels of neofunctionalization, so 16% of the time on the blue line, you're adding a random interaction after a gene duplication. You can see that not only is clustering coefficient not escaping its random equivalent, it's actually lower than its random equivalent. So to really push the model, let's choose more aggressive parameters for the number of random interactions with each duplicate. So the brown and green lines, 16 interactions, 50 interactions, and these get to biologically untenable numbers, but it's very useful to see what's happening. What you're seeing is the clustering coefficient is indeed increasing in these models, but it's unable to escape the random equivalent. Why? Because these models of neofunctionalization, adding interactions, is adding them randomly. So you're, in essence, getting random expectation as you kind of overload these models with neofunctionalization. Now let's go ahead and contrast the other plot. Again, you have the duplication and the subfunctionalization. The mechanic being is the probability that the duplication that you just made was of a self-interacting protein, so you should add a paralogous interaction. And you'll find that that has a really powerful effect. So the parameters, the probability of adding an interaction between paralogs, and even at very low probabilities, you can see that right away that clustering coefficient being measured in your theoretical network is much higher than the random equivalent network. So what are the conclusions that we can draw from this? Number of different lines of reasoning supporting the ubiquitous neofunctionalization and the evolution of these networks, and there's subtle errors in all of them. So it really draws into question how frequent this is actually occurring in these networks. Second, I think we can, at least I'd like to assert, that subfunctionalization accompanied by the duplication of self-interacting proteins is more parsimonious in these cases. And it also really highlights the importance of the duplication of self-interactions. I'll touch on that in seconds. Finally, I make it clear that I'm not denying the existence of neofunctionalization per se. It's very clear. You simply look at yeast and human, and no doubt that there have been new functions acquired throughout evolution. I do want to say, in the context of what we're looking at with these model protein interaction network evolution, that the way we're modeling it is probably not the right way, and that for those of us that are hustling these networks, we don't have to despair. I think there are a lot of mechanics that are really been underanalyzed at this point, could be incorporated into our models that would be relevant and produce neofunctionalization, such as changes in binding affinity and horizontal gene transfer, gene fusion, fission, so on and so forth. Also, numeric interactions, I believe, based on these results here, is very important to the evolution of these networks. And the very interesting thing is, almost without exclusion, any measure you ever see of network is going to exclude self-interactions. You look at the methods of any of these papers, not as I talked about any of them, and what they'll say is the first thing they do with their network is they get rid of the self-interaction so they don't get in the way for doing a proper analysis of it. I think there needs to be some measures developed to take into consideration these self-interactions and to embrace them rather than ignore them. So with that concludes my talk. I definitely want to give a big thanks to ISCB and DOE and NSC for the grant that really made it possible for me to stand up here today. And also, I have two other things I want to add. One is that my current fellowship a few months. So for those of you with labs who have postdoc, I encourage you to charge the stage. And the second thing is, if you have kids, I do this presentation at children's parties. Thanks.
So I thought it was a very interesting talk. I'm trying to kind of square what you said with what we know from the structural data. So absolutely right. These homomeric interactions are absolutely ubiquitous if you look at structures. If you look at the hetero interactions that appear to have come from homo interactions, I'm thinking of Sarah work and Pierre Leal and so on. That certainly happens, but it seems to be about 10 or maybe 20% of the time based on...
I'm sorry, if you take...
So where you've seen what used to be a homo interaction and has had a duplication and then you've got divergence.
Right. So you see the Paralogous interaction then?
Yeah, so you see this sort of thing in the proteasome, for example.
Sure.
That seems to occur about 10 or 20% of the time, and the rest of them, they're typically hetero interactions. So
right.
How does that square with the mechanisms that you're talking about? Because it seems to be the case, but it seems to be not the majority of the...
Well, I don't by any means posit the majority. As far as that, for instance, that theoretical model you saw, as far as the ability of the model to generate a clustering coefficient that far outstripped what the random equivalent is able to produce, that took in the amount of 20%, as you mentioned, to do that in style. So I don't think there's anything inconsistent there.
Okay, thanks.
Totally different point, but the fact is that the experimental techniques that we have disfavor observing that. Well, he's gone.
So, Bob was to put a rate on what he thought the neofunctionalization rate was. And obviously, you've shown why that was highly dubious. I was wondering, can you, on the basis of your results, put a new, improved, more realistic rate on neofunctionalization?
I don't have an answer to that because in the absence of this evidence showing that it's present in the network, I'm not aware of any network method which has established its rate. Now, certainly you have a whole lot of anecdotal evidence of people looking at specific proteins and their orthologs and showing the presence of new function appearing. And there are some theoretical studies. There's, and I don't know if I had the pronunciation right, Liberle and Rastogi out of University of Wyoming had a paper looking at, I believe, binding of ligands and showing that a new interaction could be formed. But, you know, they're looking at theoretical proteins, just two of them. They're not looking at a network, so I haven't seen any way to extrapolate that to come up with network-wide numbers.
Any other question? If there's not, I have one on this. It's a little bit more involved, a little bit more complicated. So assume I have a pair of proteins A and B that interact.
Yeah.
I have a pair A prime and B prime that interact, or that I don't know whether they interact. So I ask myself, at which level of similarity can I infer that the prime pair interacts? Now, the observation is that when the prime pair, A prime and B prime, are paralogs, then the probability of interaction is much higher than when they are orthogonal. I'm surprised you say sure.
Yep. Sure.
Why?
That if they're paralogs, they're more likely to interact?
Yes. The opposite of sure. Why could that be?
Well, I think if the so A and A prime are orthologous.
So again, I'm looking at my question is A and B I know interact, A prime, B prime I don't know. I have two pairs of
A and B paralogs?
A no, they're not.
Oh, okay.
A and B are not related.
Right.
It's just a pair of proteins that interact. They have nothing to do with each other than they interact. And I have a pair A and B prime. A prime is either ortholog or paralog to B to A, and the same is true for B prime.
Right.
So let's look at two. So if A prime, B prime, paralogs. If A 2 prime, B to B prime, and they are orthologs. And the orthologs are less likely to interact than the paralogs. Why?
Did you say the orthologs are less likely to interact than the paralogs?
Yes, yes, yes. It's the sure part, just the other way around. That's funny.
Data quality.
Data quality.
No, absolutely not. We looked at tons of slices of the data quality. No. This is crystallographic data, is high throughput, low throughput experiments, biochemical experiments. Nothing changes this observation.
That's weird because I have
Totally
data.
weird, yeah.
I have data showing the opposite, that the orthologs and the paralogs interact. Right? Okay.
Okay. Great.
Thanks
Okay,
again.
Thank you.